Okay, I'm going to present 3 cross-cutting projects. This means cross-cutting in that there's large-scale data generation, there's technology development and implementation, and there's also computational modeling. And as Aviv said, some of these ideas you've heard before were perhaps just packaging them in a different way. We were told to lump, don't split. So the first one is the idea of large-scale functional characterization of genes, elements, and genetic variants. So perturbations in pools of cells followed by high-content, high-resolution data. We're thinking about perturbing coding and non-coding elements, perhaps prioritized by the results of GWAS. We can think about individual perturbations, combinations of perturbations, different kinds of technologies, gene knockout, overexpression, CRISPR inactivation and activation, and also base editing to look at individual variants. And readouts can be many. We can think in terms of even biochemical readouts for coding variation, say how transcription factor variants in a transcription factor binding domain affect DNA binding, single cell profiles, cell biology readouts, tissue and cell culture organization. And different models, we can look in cells, we can look in organoids, and also do in vivo studies eventually. And the output would both be lookup tables so that you would have a bunch of data associated with individual elements or variants, but also would enable genotype to phenotype computational modeling. The second cross-cutting project is next generation Qtl studies, so better phenotypes using single-cell technology, using spatial assays, using electronic health records. So we envision sort of following up on studies like GTAC, but now using more molecular profiles, not just RNA-seq, single-cell data across tissues, histological phenotypes, maybe image data, and the subjects could be chosen, for example, at the tails of polygenic risk scores from the general population, trying to cover different developmental stages or representing disease states. And again, there would be rich computational opportunities for data mining, integration, and looking from genomic data to phenome. Okay, and the third cross-cutting project is to build transferable gene regulatory models and other multi-scale models from multi-layered data across biological systems. And the gene regulation part follows up on the very first morning from Jay Shanduri's talk of trying to predict the regulatory landscape from a megabase of DNA. We envision more global models, sort of the global regulatory consequences and gene expression output in different biological systems. And we are thinking in terms of Anshul Kundaje's suggestion of transfer learning, so trying to collect rich data, molecular data, cellular data, image data, other kind of phenotypic data, and perhaps you can collect more data in certain systems, but we want to move into more biologically relevant systems. So we can use ideas of transfer learning from machine learning, transfer from regulatory models from cells to organoids to tissues, or transfer between species. Just transferring models from mouse to human would be extremely valuable. And transfer learning could also be used to go between data modalities. So, for example, omics kind of data to, and spatial expression data, maybe to other kinds of image data. Okay.